In the last module, we took a quick look at the entire process, from sampling to delivering your biodiversity inventory results. In this one, we'll go deeper into how DNA behaves in the environment and the key factors that influence its dynamics. When we filter water at a site, we're collecting DNA traces left behind by different species. But here's the key question. How long do these traces last in the environment? Studies show that in aquatic environments, DNA traces persist only for a few hours to several days, depending on various conditions. That's why eDNA is like a snapshot. It captures which species were present at that site and exact moment in time. eDNA provides a snapshot of recent biological presence, not a historical record. Once released into the environment, DNA begins to break down. Several factors influence how long eDNA persists, including enzymes, bacteria, UV radiation, temperature, and salinity all contribute to degrading the DNA molecules. All affect how long DNA remains detectable. Another key factor is that eDNA does not travel very far under most conditions. It tends to remain concentrated near the source organism, which is why we describe it as highly localized. That's why we use integrative transects to provide a more complete and reliable snapshot of biodiversity at the site. By contrast, fixed-point sampling, for instance, can be misleading. If you only collect water from a single spot, you might miss species that are few meters away, and that can distort your conclusions. In certain environments with strong water currents, such as estuaries where freshwater mixes with marine water, or areas with significant flow, DNA can be transported away from its source. However, in most cases, detecting a species' DNA at a given location indicates that the species was present at or very near that location recently. These two characteristics have important implications. When, where, and how you sample greatly influences what you will detect. Let's now talk about two more key concepts: false negatives and false positives. A false negative happens when a species is present at a site but doesn't show up in your results. A false positive is the opposite. A species appears in your results but isn't actually there. At SpyGen, we've developed our technology to minimize both. Still, it's important to understand why they occur and how to prevent them. First, let's look at false negatives, when a species is present at the site but not detected by the technology. These can happen for several reasons, including wrong primer choice. Each primer is designed to amplify specific taxonomic groups. This ensures we can detect the greatest number of species with the fewest false negatives possible. At SpyGen, our primers are rigorously tested and proven. They represent the cutting edge of primer technology in the field. Poor sampling strategy. Sampling is a critical step in eDNA analysis, as it ensures that sufficient DNA is collected for the subsequent stages. If you collect water from a location where the species was not present, or if you do not filter a large enough volume to capture its DNA, you risk missing it. For instance, relying solely on fixed-point sampling or filtering low water volumes increases the likelihood of overlooking certain species. Biological factors. Some species naturally release less DNA. Reptiles, for example, shed much less than mammals or fish, making them inherently harder to detect. Gaps in the reference database. If a species isn't included in any database, its DNA sequence will appear as an unidentified sequence rather than a confirmed species detection. This is why reference databases are so critical for reliable results. Over the past 15 years, SpyGen has built one of the largest proprietary, field-tested, and taxonomically verified databases in the industry, and we continue to expand it every year to improve detection coverage. As for false positives, when a species is detected even though it isn't truly there, it's important to understand that eDNA detects genetic material, not living organisms directly. This is why ecological validation is essential. Two main situations can lead to detections requiring validation. DNA from contamination sources. If proper field protocols are not followed, DNA from outside your study area can contaminate samples. For example, sampling equipment previously used at another site without proper cleaning, or collecting samples from vessels that transport fish commercially. This is easily prevented by following proper field protocols, such as using clean, dedicated sampling equipment and applying proper handling procedures between sites. This training ensures you know exactly how to avoid these issues. Also, natural DNA transport. eDNA can move with water currents, which is actually informative for understanding connectivity. For example, in estuaries where freshwater mixes with marine water, you might detect freshwater species DNA in coastal samples, revealing the influence of river outflow. In areas with strong currents, DNA might travel from upstream locations. This is why our experts conduct ecological validation on all results, flagging detections that don't match expected species distributions for your sampling environment. These contextual detections actually provide valuable information about water movements and ecosystem connectivity that needs to be taken into account when designing the sampling strategy. Another factor to keep in mind is season. During reproduction, many species release more DNA through gametes and reproductive behaviors, making them easier to detect. For monitoring programs that track changes over time, sampling during the same season each year ensures differences reflect actual biodiversity changes rather than seasonal variation in DNA shedding. Our comprehensive approach helps reduce both false negatives and false positives. Our protocols are optimized for different situations, and our experts will support you in identifying the most appropriate one for your specific project needs. Large volume integrative sampling ensures sufficient DNA is collected across multiple microhabitats, significantly reducing false negatives. Using 12 PCR replicates increases the probability of detecting rare species. High sequencing depth provides the statistical confidence needed to distinguish true detections from sequencing noise. Finally, expert ecological validation ensures that specialists review the results for ecological coherence before delivery, flagging any detections that may require contextual interpretation. By understanding what can affect eDNA results, you can minimize risks. Good field practices are the foundation of reliable results. Follow sampling protocols precisely, maintain strict decontamination procedures, and use only single-use materials between sites. Store samples properly with the conservation buffer, and document all conditions thoroughly, including location, time, weather, and vessel activity where relevant. Whenever needed, our experts will support you in designing sampling protocols that minimize error risks for your specific project context. This brings us to the end of this section. You can now move on to the next module.